Welcome to the approximately 200 people joining this deep dive into how Mass General Hospital's Center for the Neuroscience of Psychedelics will leverage MGH's cutting-edge neuroimaging technology to better understand the ways that psychedelic compounds facilitate lasting changes in mental health and well-being. For those who are new to this series, I suggest you also listen to the first Salon recording for an overview. Today, you'll get a behind-the-scenes look at one of the key pillars of the center being brought to life at MGH, Harvard Medical School's primary teaching hospital and hub of the Boston biomedical ecosystem. I'm Dick Simon, chair of the center's advisory council. I spend all of my time doing what I can to advance psychedelic therapies for treating a wide range of mental health issues. I'm motivated both by the astounding data supporting the efficacy of psychedelic therapy and from having experienced the seemingly intractable nature of mental health issues in my own family. I'm excited about this center with its unparalleled human and technological resources, as well as its integrative approach to psychiatry, chemical neurobiology, and of course, neuroimaging, the subject of today's salon. As a reminder, at any point you can type questions into the Q&A button and we'll address them during the question and answer session. I'm now happy to introduce my good friend, Dr. Jerrold Rosenbaum, founding director of the MGH Center for the Neuroscience of Psychedelics. Jerry is psychiatrist and chief emeritus at MGH's psychiatry department, which he stewarded as the top-ranked psychiatry department in the US for 20 years. He's also professor of psychiatry at Harvard Medical School. Jerry brings 47 years experience as one of the preeminent psychiatrists and psychopharmacology experts in the world. It's my honor to introduce Dr. Jerrold Rosenbaum. Jerry.
Thanks, Dick. I appreciate your introducing the webinar and for all you're doing in this space, both for the world and for the Mass General Center for the Neuroscience of Psychedelics. As Dick mentioned, this is the first in a series of deeper dives into components of the Mass General Center for the Neuroscience of Psychedelics. I know many of you joined us last month for the comprehensive overview of our center, and today we will provide a more granular view of the potential of neuroimaging in the quest to explore and understand how psychedelics change the brain. If you missed the initial webinar, we are happy to send you the link where you can be introduced to the center's origin story and its four main program areas. Those four include the Chemical Neurobiology Lab, along with the Schultes Legacy Project for Psychedelic Discovery, through the Harvard Archive of Schultes' amazing Amazon exploration. That will actually be the subject of our next webinar, so save the date, September 15th. I think you'll really get a kick out of it. Second component of our center is the group that does the clinical and translational human research studies that you also heard about on the first webinar led by Dr. Sharman Ghaznavi. And our Education, Dissemination, and Training Division, which is powered by our Mass General Psychiatry Academy and its 65,000 professional members. And finally, in today's focus, the Martinos Center for Neuroimaging, which, as you heard last time, employs more engineers than MIT and pioneers developing the tools that define the field and can image from the molecular to the network level to deconstruct how the brain allows us to think, feel, and behave. When I entered the field of psychiatry in 1973, the main tool available to address psychiatric suffering was a trusting therapeutic relationship. Even though we knew that mind and brain were one, we had a limited window on how the brain gave rise to the mind and how experience can change the brain. Given the burden of psychiatric suffering, as we say here, no family goes untouched, the need for insight into how the brain can heal and change was and is compelling. Genetic risk profiles, developmental injury and trauma, life events of all kinds, neuroinflammation, environmental toxins, and so much more all conspire to create risk for psychiatric disorders. And yet we know that even a dysregulated brain can change and heal through a variety of processes, biological and psychosocial, a change subsumed under the concept of neuroplasticity. We have reason to be very hopeful that discovery in the field of psychedelics will lead to new generations of therapeutics and assisted therapies so that we can make less and less use of the term treatment resistant when speaking of those who suffer. When we learn more about what's happening in the brain during a psychedelic experience, for both patient and therapist, individually and collaboratively, then we will apply that knowledge to develop new treatments and take the often long and distressing trial and error approach out of the therapeutic process, so patients may get the best treatment for themselves right out of the gate. Today, you'll see an introduction to how one might actually image the therapeutic relationship, so key to therapeutic success when treating a human being. We are really pumped to have brought together this exceptional team to answer the question of how psychedelics can change the brain and deliver us new and better tools to heal the brain. So sit back and continue or join our journey with today's focus on neuroimaging. And it's a great pleasure to introduce my spectacular colleague, Dr. Bruce Rosen, who is a professor of radiology at Harvard Medical School and directs the Athinoula A Martinos Center for Biomedical Imaging. His team in the early 1990s developed fMRI and continues to develop new tools that are state of the art for helping us to actually probe the brain in a way that was unimaginable before. So I'm pleased to introduce Bruce, whose brilliance is only exceeded by his humility, Dr. Bruce Rosen.
I don't know if the tone on these cameras picks it up, but I'm blushing severely from that very generous introduction. Let me share my screen. Oh, there we go. Hansen, the first of the deep dives, as Jerry mentioned, in this case focused on the role of human neuroimaging. So the first question is, why is it that we're so excited about neuroimaging? What are the unique properties of our ability to image the human brain that we think will make an important, though not singular, contribution to our understanding and development of the role of psychedelics in treatment of a wide variety of disorders? So by way of analogy, let me present something that I'm sure is very familiar with you. And that's this analogy between the human brain and a computer. And it's both the similarities and, I would maintain, differences that really motivate our interest in neuroimaging. So let's think about a digital computer. One of the interesting things, of course, about this thinking device is the many different scales that you would need to understand to really understand comprehensively how it's working. Everything from the very detailed quantum mechanics of P and N junctions within the semiconductors, the material science of those, the different components, transistors, integrated circuits, et cetera, how they connect at a circuit level, a local circuit level, how the wiring between those different components connects to the whole, and of course to the overall operating system of the computer itself. And of course these days, equally important, how individual computers all talk to each other within the context of the broad networks that we use so routinely and that you're using as you're watching this talk today. Now, a human brain can be understood at very similar levels. Everything from the molecular level, studies of metabolism, neurotransmitters, extremely important as we want to understand the role of psychedelics, synaptic activity, the dynamics of the receptors, how they're locally connected, how electrical activity within the brain is generated, both within single cells and local networks of cells and this local network wiring, the broader sense of network function, distributed function across the entire brain that we now know from 20 years of functional imaging, the dynamics of that, the connectivity of these networks, And then, of course, finally, how brains interact with each other, the sociology of how individuals all interact with each other. Each of these, of course, is another level of understanding of the human brain. But I would and one of the wonderful features of human neuroimaging is that we have unique tools that allow us to address each of these different levels. This is really the power of the tools that my colleagues at the Martinos Center, and of course others around the world, have generated over many years. So for example, we can use positron emission tomography or PET scanning to directly image metabolism, neurotransmitters, and receptor dynamics. We have tools to see the electrophysiology in the brain. For example, magnetoencephalography, kind of the more expensive and much more detailed cousin of the more familiar EEG. We can use that to see local circuit functioning and distributed electrical activity. Network function can, of course, be seen using tools like fMRI that Dr. Rosenbaum alluded to, and other tools like our tools for diffusion imaging we can use to see connections in the brain, so-called connectomics. And finally, as you'll hear about from Dr. Napadow's talk, we'll be able to see how different brains actually interact with each other, to watch this process of interaction, which we know to be such an important component of our therapeutic efficacy in treating psychiatric illnesses. The biggest challenge, and I think the biggest difference, however, between, say, how we want to understand a human brain and how we might understand a digital computer, is that to really tackle the most challenging issues that we have today in the human brain's problems like depression, substance use disorder, chronic pain disorders, we really need to see the brain across all of these levels. It's not sufficient just to understand the molecular level if we can't see how that then changes to longer-term plasticity across networks and how individual brains then can interact, say, between a patient and a therapist. The beauty of our neuroimaging tools is that we can image across all of these levels and do so, of course, in a living human subject and in their therapist. So with that brief introduction, let me introduce our two speakers today who, in their work and special expertise, really span all of these scales that I've just presented to you. The first of our speakers will be Hanne Hansen. Hanne comes to us from Copenhagen, Denmark, came to us as a graduate student, has stayed as a fellow, and now has become a faculty member within the Martinos Center. And in her relatively short scientific career, has already become one of the world's experts in a unique technology that we have at the Martinos Center, and that's the integration of PET imaging with MRI. And you'll see the value that these combined technologies bring together. After Hanne, we'll introduce Vitaly Napadow, but I'll get to him in a bit. So with that, I'd like to turn it over to Hanne for her presentation. Hanne?
Wonderful. I will also start with sharing my screen. There we go. Thank you, Bruce, very much for that very kind introduction. I am very excited to be speaking here today, honored as well. I think it's a very fascinating aim and topic, of course, my interest. I will be presenting to you some examples of neuroimaging from research groups around the world. That in itself is super exciting because it shows us how much interest and potential there is in these psychedelics. So a few examples from the research of how psilocybin and MDMA might work in the brain. I will start out, oops, sorry, just gotta speak this word. Sorry about that. I will start with a little bit of an overview and emphasize again what Bruce just said, the importance of looking across different levels to understand how these drugs work. At the very global level, we're talking about connectivity, how different areas of the brain represented with these small dots are connected to each other. After a treatment with a psychedelic drug, we see that this connectivity is increased. We simply have more connection across the brain. At the middle layer, we're talking about entropic brain activity. So in a normal state, the brain will have this really rhythmic and very predictable way of being active. But during a psychedelic drug experience, there might be a higher diversity of signaling, perhaps more disorder, and that disorder is something that we can interpret as higher entropy. If we deep into the cell levels of the brain, the lowest level, very specifically, some cells will become more active when we give this psychedelic drug. I'll give you some examples of how we get into this, but first off, I will give you the briefest of introduction to this positron emission tomography that Bruce also mentioned in his introduction. It is an imaging technique where we have molecular probes that are really specifically binding to a target that we are interested in. A protein, a transporter, an enzyme in the brain or in the rest of the body. Because this molecular probe is radioactively labeled, our scanners can detect where that radioactivity is, and thereby we can find where our target of interest is. This enables us to create atlases or images of where different components of these neurotransmitter systems are. This is an example of the serotonergic serotonin neurotransmitter system, which is really important when we're working with these psychedelics because they bind to different components within this system. So here we have 4 different receptor atlases and 1 transporter. This enables us to look at how these drugs interact with each of these different components, one at a time, and figure out some details. First off, I will be focusing on the serotonin 2A receptor because that's the main target that psilocybin binds to. So this is an image that basically represents the same thing as the previous slide, how the 2A receptors are located throughout the brain. If you give psilocybin, these 2 drugs will compete, so the molecular probe and psilocybin will compete to binding to that receptor, so we will have lower binding of the molecular probe, these subdued colors. Now in this study, the subjects received different doses of psilocybin, and you can see how the intensity of the experience differs in intensity, but also over time. Now, a really important finding here is that there's a really strong association between the intensity of this experience and how much psilocybin is binding to that receptor in the brain. That's what we call occupancy. Now, so this established the relationship between the drug dose that we're giving and the target engagement in humans. And that might be really valuable in determining the best therapeutic dose for patients. We don't want to give too much, we don't want to give too little. We'd like to hit that sweet spot where we help our patient populations the most. And studies like these can help us do that. Turning to a different imaging technique, or is called functional connectivity. It's an fMRI technique. And it basically shows you the association in time between different regions. So here you have region A and region B, and you see how they nicely fluctuate together. These are really strongly connected or high functional connectivity. Now, throughout the next couple of slides, you will hear the network name default mode network plenty of times. This consists of 4 different anatomical regions in the brain, some in the front and some in the back. This default mode network is especially active when we are at rest, when we're asked to do nothing. And psychedelics really affect this default mode network activity, or connectivity, if you will. So this is an example of how they're doing that. Again, this has been replicated across different research groups in the world, Franz Vollenweider and also Robin Harris. So you see the decreased, the blue colors here, connectivity between the front and the back of the brain. A different way of representing that is by imaging multiple times throughout the psilocybin experience. So this is the connectivity before you give psilocybin. You have the strong connectivity with the front and the back of the brain. Now, 40 minutes after the administration of psilocybin, you see how this high intensity coloring in the back has disappeared. And it takes a while before that comes back. It's not until 5 hours later that you see some of the activity or connectivity coming back. But these results have all shown you the connectivity during the psilocybin experience. What about the day after, the week after, or the month after, where the drug is no longer in our system? Well, that's something that, again, Robin Carhart-Harris, which is one of the pioneers of neuroimaging psychedelics, has investigated. He did a clinical trial in treatment-resistant depressed patients where he measured this functional connectivity the day after psilocybin treatment. And what he saw is that there's increased connectivity, again, in this default mode network. So the integrity of this network is now normalized, and he also saw that there were improvements in mood. And that is ultimately what we're looking for. A treatment-resistant depressed patient is a very vulnerable patient population, and one that we would like to do more for. We'd like to advance our knowledge in our treatment of these, like Dr. Rosenbaum talked about in his introduction. Another way of sort of putting words on these results is that it works, psilocybin may work as a reset mechanism. It allows the resumption of normal functioning, or it's like finding an equilibrium between order and disorder in the brain that allows us to be healthier and well-thinking. So we begin to understand how changing the brain networks is associated with disease improvements. And understanding disease improvements is one step towards understanding the disease itself and helping the patients. Now, at the second and third level of that first schematic that I showed you, we're now back at the entropy. So in normal state, we have rhythmic and ordered activity, whereas during a psychedelic drug, you would have a signal diversity, or could also be said as higher disorder. Again, another word for that is high entropy. We're again seeing that in the front and in the back of the brain. And when this group, we're looking at what specific cells in the brain, now we're at the bottom layer of the levels, are actually mediating such effects. Well, those cells are called pyramidal neurons, and those are the cells that have this serotonin 2A receptor localized. So with such a study here, we're identifying changes in synaptic activity, which leads to changes in electrophysiology with the MEG here, and changes in network function. So remember back to also Bruce's analogy, how it's really important that we look at different layers. Well, we're doing that in studies like these, and we're piecing together the puzzle between synaptic changes in the brain and the sociology or the well-being of patients. Now, turning to a different psychedelic, but equally exciting and important, is MDMA, that has been particularly promising in the treatment of PTSD. We're really thankful that MAPS is doing such a great job in promoting the research of MDMA and associated psychiatric diseases, and also fortunate that we get to collaborate with them. MDMA doesn't really affect the serotonin 2A receptor as much. It has a different target within this neurotransmitter system. It binds to the serotonin transporter particularly. And the effects are long-lasting. So if you've taken MDMA, this serotonin transporter binding remains decreased for quite a while. So perhaps there's some shared mechanism, because also psilocybin also have long-lasting effects on our well-being and personality, as many people have shown, and I'm sure you might also have heard of. When we have lower serotonin transport in the brain, we would also have lower functional connectivity, which this graph shows us. It also shown in the blue color here on the brain atlases. This again is perhaps a shared mechanism between psilocybin and MDMA, because they both acutely decreases this functional connectivity in the brain. Shared mechanism is something that we can learn of. All of this adds to this big puzzle of how the psychedelics work in the brain and which psychedelic might be beneficial for which diseases. In summary, psilocybin and MDMA are being positioned as some very powerful therapeutics for treatment-resistant depression, PTSD, and other psychiatric disorders. But we're only scratching the surface. I've shown you some really, really promising early results, but we need larger clinical and rigorous studies to confirm these early and exciting data. If we combine clinical studies with neuroimaging, it gives us insight into how these psychedelics alter the brain. Again, the more we know about the treatment, the more we learn about the disorder, and the better we can help these patients, which is our ultimate goal. One of the things that I showed you was that the increased connectivity in patients after treatment with psilocybin may be a way of how the brain escapes from its usual tightly constrained and predictable way of working. A really exciting study that we're doing within this Center of Neuroscience of Psychedelics is Dr. Ghaznavi's investigations of how psilocybin might change rumination in depressed patients. This is the negative thought process that really affects a lot of the depressed patients, and we hope that psilocybin might be able to take them out of this negative thought process. This, of course, we're combining with our super high-tech fMRI scanners. We look forward to getting the project started and seeing the results, of course. If we combine data from different imaging modalities, we will have more detailed mechanistic information how different psychedelics work. And this is really where the MGH is at the forefront of the research. We have the technology with these simultaneous PET-MR scanners, as Bruce mentioned, to really push the boundaries of this research. We keep developing the technology. So we hope to be innovators. We hope to be at the front of the research and confirm these early studies that I've shown you, but also bring the research much further and ultimately hope that we can bring these psychedelics to many patients and help them. There's lots of questions that we could still answer. The functional connectivity that we're seeing, how is that associated with synaptic density? So how the brain is sprouting and communicating with itself. This is something really new within the positron emission tomography field because we now have these molecular probes to actually study it. Is the psychedelic experience necessary for the therapeutic effect of psilocybin, for instance? There's a lot of things that suggest yes, it is, but we don't have hardcore proof of that yet. So we need to study this in further detail, and Dr. Vitaly Napadow will, in his presentation, give examples how that might actually be done. So with that, I would just like to thank you for your attention. I look forward to any questions that you may have, and now I'd like to give the word back to Bruce. Thank you.
I hope everybody was as excited as I was. I hope everybody was as excited as I was that you pretty effortlessly scaled across 9 orders of magnitude, you know, from nanoscale synapses all the way to whole brain networks and back again. It was really very elegant. Thank you so much for that. So next I'd like to introduce another one of my colleagues who's been at the center for quite some time now, Vitaly Napadow. Like many people at the Martinos Center, Vitaly is really a polymath that comes with great expertise in a wide array of disciplines. He's a PhD biomedical engineer and a licensed acupuncturist. He's done some of the foremost work in looking at alternative and complementary treatments like acupuncture to study their effects on the central nervous system. And over the last 10 years has developed a comprehensive program in studying the effects of chronic pain on the brain. I was inspired to bring Vitaly to this based on several questions that came up at our first salon. People were asking about the nature of the therapeutic interaction between the subject and therapist during and following the course of a psychedelic treatment. And it's in fact the tools to be able to directly see this interaction, to watch the interaction, not just in the patient's brain, but how that patient's brain is interacting with the therapist's brain and vice versa. That's another one of the domains that Vitaly has been a pioneer of. This is something called hyperscanning, and you hear more about that. Vitaly is relatively new to the field of psychedelics, but not new at all to the use of advanced neuroimaging tools to study the brain. And I think you'll find that this particular facet of how we can study multiple brains at once will be especially relevant to how we want to move the field of psychedelic research forward. So with that, I'll turn it over to Vitaly, but with a reminder before I do, that please type in your questions into the question and answer slot. We'd like to collect them, and then Dr. Rosenbaum and I will be sure to field them at the end of the session. So type in your questions, and with that, I'll turn it over to Vitaly.
Great. Thank you very much, Bruce. Hope everybody can see the talk. We can get going. So it's a real pleasure to be here to present to you all. And as Bruce mentioned a second ago, I'm here really to talk about more from the standpoint of the nature of the therapy and the guided therapy that's associated with psychedelics. So the idea here is that psychedelics might be important to induce this very plastic state in the brain, such called neuroplasticity, wherein you're able to learn, the brain is able to learn, clinical patients are able to recover better, so that when the this high neuroplastic state is combined with some sort of therapeutic intervention, such as guided therapy, we're able to get enhanced clinical outcomes. And so what I'm going to be focusing on specifically is how do we investigate this process over here? So yes, psychedelics are inducing some sort of brain changes or having an effect on neuroplasticity, but then what happens? How does this guided therapy then combine with this neuroplastic state from psychedelics in order to create beneficial clinical outcomes? And my interests in this research has been to try to understand therapeutic alliance as a key element of clinical therapies. And so positive social interactions engender therapeutic alliance between a patient and a clinician. And it's known from many clinical studies that this enhanced therapeutic alliance can in fact significantly impact depression, pain, and other clinical outcomes. So the tool that we've been using is something called hyperscanning, and hyperscanning is a fancy word, but really it just means that you're synchronously imaging more than one individual. A lot of this work has been done with technologies such as EEG or electroencephalography, and this has been applied to all kinds of different contexts, such as interaction between a pilot and their co-pilot, how people learn how to juggle over time, and how that changes the brain and brain concordance, and also with musical performance. And as you see here, I didn't mention anything about the clinical interaction because there's been so few, if any, studies that have applied hyperscan neuroimaging to really try to understand the relationship that's formed between a patient and a clinician. Now, in addition to EEG, you can also do what's called fMRI hyperscanning. And so fMRI hyperscanning allows you to assess brain-to-brain communication between individuals on both a cortical and a subcortical level. So that's important. Also, fMRI has improved spatial localization compared to EEG. So there's many reasons why we might want to engage in fMRI hyperscanning as well as EEG hyperscanning. Now, the question is, how do we actually do this? There have been coils that have been created where you can fit two people into one scanner in order to image synchronously both the brains of the two individuals. But I would argue for our sort of clinician-patient dyad, this is probably a bit too close for comfort. And so we didn't go in this direction when we were doing our study. In our study, we had two synchronized functional MRI scanners. And so this is a real advantage of the Martinos Center, where we have, I think, ballpark 8 human fMRI scanners. So there's many scanners to choose from to do this kind of research. And fMRI hyperscanning is actually, as you'll see, is very technologically difficult. There's a challenge there. And very few centers in the world are even engaged in this kind of research. I would say we're probably one of 4 centers in the entire world who are doing fMRI hyperscanning research, and the only one who are studying a clinical interaction, clinical context between a patient and a clinician. And so our hypothesis is that brain concordance in social mirror circuitry areas, such as ventrolateral prefrontal cortex, anterior insula temporal parietal junction, will be linked to therapeutic alliance and analgesia in a clinician-patient interaction. And so in our study, while we're here very interested in psychedelics and the role of guided therapy, in this particular study, we were studying chronic pain patients and acupuncturists and not psychiatrists. However, patients were brought in for this study. They had both. We directly compared a scenario where there was a clinical interaction and a clinical intake, and this was followed by an MRI, a hyperscanning MRI with a social interaction, versus when the patient would meet a completely different acupuncturist and there would be no interaction, and the order was randomized for the sake of this experiment. And so our setup involves 2 MRI machines. We have MR compatible video cameras, MRI compatible audio microphones in both scanners. The image is taken of the entire face. We've figured out a way to get the entire face into the image, which is then transferred over to a video screen for the other subject. So each subject can see the face of their interacting partner, which is very interesting. And both scanners are controlled by laptop computers, so we can directly synchronize the imaging in one scanner with the other scanner. And we monitor this regularly because we're doing it over the network to make sure that the delay between the 2 scanners is under roughly 50 milliseconds. And at a 50 millisecond delay, you will not notice any noticeable lag in the interaction between one subject and the other. So that's important too. And in our particular study, we had the patient that was undergoing an evoked pain task where we have a cuff wrapped around their gastrocnemius muscle, inducing a pain stimulus. The patient is able to rate their pain in response to this stimulus, while the acupuncturist is able to control with a button press a device that is producing electroacupuncture therapy to the needles, to the patient. And so the reason we actually chose this context for our particular study is because it's very ecologically valid. This is something that acupuncturists directly do, is pressing buttons to control some sort of therapy through the needle. However, it's important to note that this kind of context can also be exported out to study other types of interactions with other types of therapists, including psychiatrists with mental health patients. We're able to collect not just bold fMRI data from the brain, where we can look at the patients being treated and the clinicians that are doing the treating. We also have physiological monitoring for autonomic signals such as cardiac, respiration, and skin conductance. And importantly, we also can record the video image of the patient and of the clinician while they're engaged to see what the other one sees, to see if there's any sort of transfer there. And we use artificial intelligence algorithms for automated face recognition and emotion detection to do this. So I'll show you in a second. So one of the things that we found in our pilot study is that even though we are inducing the same amount of pressure in all cases for treated versus non-treated conditions, when the clinician is treating and the patient thinks that they are being treated, the pain rating that they give is significantly less than if they believe they are not being treated. Also, the pain that's rated by the clinician as to what they think the pain is of the patient, this is something that we call vicarious pain, is also reduced as rated by the clinicians. And what was, I think, really interesting is that there was a correlation between the two. So the more that the patients report an improvement or reduced pain in response to the therapy, the greater the ratings of the perceived efficacy in terms of reduction in vicarious pain by the clinician. So something is happening here where the clinicians are able to rate improvements or reduced pain ratings when they're actually doing the treatment. And so the question is, how might they be doing this? How can clinicians accurately perceive the efficacy of their therapy? Maybe it's through the video transfer of the facial expression, for example, because that's the feedback that in this particular experiment they're able to receive. And what we found is that in fact might be the case, that there's actual mirroring that's happening in the change of the facial expression for treated versus no treated in the part of the patient versus on the part of the clinician. And this is across all kinds of automated emotion detection in both the patients and the clinicians using the videos that we have recorded. But what was, I think, even more interesting is that when we looked at individual dyads and differences between individual patient-clinician dyads, we found that there was a relationship between the mirroring and both the therapeutic alliance that's rated by the patient. So the more the mirroring, the greater the therapeutic alliance as rated by the patient. And also it was linked to analgesia, the difference in pain response in the patient for treated versus non-treated trials was greater. They reported greater analgesia when there was overall mirroring by the clinician to the patient. So the more that the clinician can mirror the effect in the patient, the greater the clinical outcomes, the better the clinical outcomes. Now, why might that be? So why might clinicians mirror their patients? Well, the clinicians are trying to improve the clinical outcomes in the patients, right? And we know that also from a very early age, babies learn to imitate the facial expressions of their caregivers in order to upregulate therapeutic alliance, in order to upregulate empathy from the caregiver. Babies need this in order to survive. So speculatively, does clinician-patient mirroring also reflect therapeutic alliance and empathy? And is it supported also by brain concordance in specific social mirroring circuitry? And is this something that could potentially be upregulated by psychedelics? So if placing the patient into a hyper neuroplasticity state, are we able to then engender greater mirroring in terms of facial expression and in terms of brain response? So in our case, we were able to track brain response by looking at the trial-to-trial variability in the response in the patient. So this is looking at a particular area of the brain called TPJ, or temporal parietal junction. This area has been strongly linked with theory of mind and with empathy processing. And what we found is that the more that area is activated by the patient, the more you have activation in the clinician in specific brain regions. And those regions were upregulated for the social interaction condition versus the no interaction condition. So if they've built a relationship with the clinician by having the social interaction beforehand, there is greater concordance in the brain in response to these trials. And that's the case for a whole cohort of brain areas responsible for social mirroring, not just TPJ, but also ventrolateral prefrontal cortex and anterior insula. But what was really interesting is that it was only in the TPJ where greater concordance, greater brain concordance between the patient and the clinician was associated with better analgesia. So the clinical outcomes were better if there was greater brain concordance between the TPJ of the patient and of the TPJ of the clinician. You know, whether this is also the case within a psychedelic experiment with an effective cue with say a depression patient, that I think remains to be seen and would be really interesting to study. So we've also been able to port this kind of experiment to an EEG hyperscan environment, and we have a different group in my lab that's working on this. And so here we can have a patient and a clinician inside of a sealed room with one-way mirrors where we can monitor them, and we can track, you can see here the nets that are tracking EEG activity in both the patient and the clinician while they're interacting. And this is really important because we might be able to look at what's happening during the interaction, not just what's happening as a consequence or a result of the interaction, which is more the design of the study that we did with fMRI. So here we can actually collect brain data while they're interacting and engaged in talking therapy. So here what we found is that we actually trained an augmented versus a limited interaction condition in the part of the caregiver, the clinician, and we found that there was greater internode density, but only for the augmented condition with the treated trials compared to the non-treated trials. And so this is really interesting pilot data, but something that we're actually in the middle of the study and hoping to collect more data. So the idea here is, can we now port this kind of experiment, which I just showed you in a pain research context, into the kind of context with a guided therapy in conjunction with psilocybin or some other kind of psychedelic? So here, for example, we might want to focus on different brain areas, not just the social mirroring circuitry, but maybe the relationship, for example, the social mirroring circuitry in the clinician to brain response in the patient in a different set of brain areas. In this case, for example, areas that we know to be coded to elements of rumination, such as ventral ventral PCC, or posterior cingulate cortex. And this is in a different study from our lab, where we actually had chronic pain patients engaging in what's called pain catastrophizing, of which rumination is a very important part. And the more that they ruminate, and the greater this rumination score that they then self-report, the greater the brain response in this particular part of the brain called the posterior cingulate cortex. So knowing this, can we then look at this specific brain area in an MRI hyperscanning experiment to see if this is related to brain concordance with the clinician in a guided therapy experiment when the patient is undergoing a psychedelic experiment. So in summary, Hyperscan, fMRI, and EEG can be used to study brain mechanisms supporting this dyadic patient-clinician interaction and looking at therapeutic alliance. Also, the psychedelic experience might change how the patient relates to the clinician and how the clinician interacts with the patient, potentially enhancing therapeutic alliance and clinical outcomes. The idea here is that we really want to establish the importance of guided therapy in psychedelic medicine. And that is something that heretofore has been really difficult to do with social neuroscience in general, because there's been so little work that's done in clinical context with this type of social neuroscience experiment. And there's a series of questions I leave for you up here to ponder and maybe to discuss in the panel. And I think in the interest of time, what I'll do is I'll turn it over to the questions and to the panel and your Q&A. So thank you very much for your attention. And maybe I'll just leave up the slide right now. And I'm happy to answer any questions. Thank you.
Jerry, you might need to unmute. There you go.
Okay, we have a handful of questions from the audience. We're probably not going to have time to get to all of them, but we will email you the answers to those. So I'll just pick a couple. One for Hanne, which is, let me see if I can find it again. It said the day after the drug administration, the drug will no longer be present in our body. So the effects we see are indirect or secondary effects mediated by psilocybin. Can you say more about that?
Here? Yeah, a great question. Thank you. So what we call the half-life, the time that these drugs are actually active in our body is not very long in sort of a medical perspective. They probably around for about 8 hours. So whatever effects we see the day after are indirect effects. These are effects that are sort of started by the drug itself, but the brain is sort of actively starting and initiating.
And a question for Vitaly was about whether we would expect to see different concordance with the type of therapist and type of therapy training, like a Freudian versus maybe a behaviorist or something like that.
Yeah, this is something that clearly has not been explored. Now, interestingly, different types of therapy in clinical trials have not resulted in significantly different effect sizes in terms of clinical effects. But that doesn't necessarily mean that that's going to be the case on a neurophysiological level. And so it might be the case that when psychedelics are involved, there would be differential clinical effects. And if we better understand the mechanisms underneath that and using these kinds of neuroimaging techniques to better understand the mechanisms, we might be able to better pair certain types of guided therapies with certain types of psychedelics. I think because psychedelics have such a strong effect on the brain, for something like this type of context, it actually might be very interesting to look at different types of therapy.
So another question, which would be relevant to eventual wider spread therapeutic use, was whether there have been any studies of younger people like adolescents, and also any studies of microdosing that might reveal either as follow-on from a standard dose or as a treatment itself, whether you see similar effects short or long term.
Hanne, do you want to take that one?
Oh, I can certainly, I don't know too much about the adolescent studies. As far as I know, I don't know any studies going on with psychedelics in adolescents. But the microdosing is a really interesting phenomenon and something that is getting a lot of attention around the world, both with clinicians, but also just in the general public. So far, it's been really hard to sort of establish what is a microdose. Does it mean that you don't hallucinate, or is there just a bit of hallucination? Certainly, microdosing would be easier from a clinical perspective because you might not have to engage a therapist in all of the sessions. So if we could identify a microdose, that would be a really good way forward for these psychedelics. I don't know whether there are any neuroimaging data with a microdosing. There certainly should be something coming up just due to the general interest there is in it. But nothing published as I know that I can sort of reference to you guys.
Okay.
I'm wondering, maybe Jerry, I could jump in just for a second, and Hanne with a follow-up. You had mentioned briefly in your talk the notion of the role of the psychedelic, hallucinogenic experience itself, and whether that's essential to these changes or not.
Yeah.
In the same way that we've discussed questions around microdosing, is there a way that we could potentially study a range of small molecules of the kind that we'll hear more about in next session's talk from Steve Haggerty, but
Yeah.
look at their effects on the brain, these changes in entropy, changes in distributed connectivity, to really try to understand exactly what the role of the hallucinogenic experience versus activation of these receptor systems potentially without that, and what that therapeutic effect might be?
Yeah, absolutely. It's a super interesting research field, and especially with Steve's knowledge and his connection with his large library of plants and sort of the active drugs inside the plants. There's a huge potential there that we could study these. And because these are plant materials, it might be easier for us to go into a clinical population. Let's first start with healthy volunteers to study how these plant products might affect our brain, how hallucinogenic they are, and how compare them to how psilocybin works, for instance. Another way of doing this is to do a more chemical or synthetic way. So there are libraries around the world with compounds that all bind to this serotonin 2A receptor, but they're not all hallucinogenic. So these are sort of molecular biology mechanisms that differ between all of these drugs. And we can start out by studying these in animals first, because these drugs are not for clinical use yet. But absolutely that would give us some really nice mechanistic information that would drive the research forward and also pinpoint us to the best way forward for the drugs and patient populations.
Well, I apologize to the questions we haven't addressed. Again, we'll try to get answers out to you, but that was a wonderful question to end on, because it points to our hope and for the future of this enterprise. And let me turn it back now to Dick Simon to say a few words, and then I'll come back on to for a closing remark or two.
Great. Well, thanks to Bruce, Vitaly, Hanne, and Jerry for sharing your insights today. This is just like amazing, and I think conveys the level of excitement and the potential. So I also want to thank everyone who's joined and listened and asked great questions. Before we wrap up, I just wanted to highlight one thing, that unlike other areas of medicine, at this time, there's almost no government funding for research related to psychedelic therapies. Now, this will definitely change over time, but now is the moment when philanthropic dollars and support can and will have the greatest leveraged impact advancing the field of psychedelic treatments for mental health. So thanks to the generous foundational donors, there are several studies that we're launching at Mass General, with many more in the pipeline. But I just wanted to make sure that people sort of realize this is the time or the moment where any support as we're growing this center or whatever else you may be involved with is so critical and so leveraged and so valuable. So again, thank you to everyone for joining, and let me turn it back to Jerry.
Dick, thank you for being our patron and supporter, and for your work bringing this effort together. I want to remind people that the next webinar, September 15th, is Steve Haggerty. You'll hear about the Schultes legacy project that we've alluded to several times, one of the components of our chemical neurobiology division of the center. That would be really cool. You'll really enjoy that. And I hope you will, as Dick said, stay with us on this remarkable venture. We're early in this journey, but it's already looking very exciting and promising. And we look forward to having you on our next webinar and into the future. So thank you again for joining, and have a wonderful rest of the day.